Hello students, it's Dr. Sansom here for the pre-lab video for experiment 10, the determination of water hardness by titration. In this lab you will have a process skills quiz, but it is not specific to this lab. It's more about your experience with process skills over the course of the semester. So in this experiment you're going to be using a titration, and it's a little bit different than the titrations that you've done before, which were acid-base titrations. This one is called a complexometric titration because we're going to know that it's done when we've completely complexed the metal ions, calcium and magnesium, that are in our hard water sample. So magnesium and calcium will bind to our indicator, which is called Calmagite, and when they're bound, the indicator is red. But when the indicator is released, because the magnesium and calcium ions are bound completely to EDTA, which is what you're adding in the titration, then the indicator is released and it will appear blue. So you'll be able to see this clear color change as soon as you have added enough EDTA to completely react with all of the calcium and magnesium that is in the solution. And of course, calcium and magnesium are the things that cause water hardness. One of the things that's related to this experiment is the idea of coordinate covalent bonds and ligands. And EDTA is a chelating ligand. This is the molecule EDTA here drawn with like a line bond form. So each corner here is a carbon atom and it's saturated with hydrogens if they're not shown. And then we have our metal ion in the middle, and notice that each of these atoms on the EDTA molecule that is making a bond with the metal has a lone pair. Like nitrogen would have a lone pair, our O- has 3 lone pairs, but it has at least 1 lone pair that it can donate here in this ligand-metal bond. We call this a coordinate covalent bond. It's a little different than a traditional covalent bond because usually you have 1 electron from each of the 2 partners that are making the bond, but in a coordinate covalent bond, both of those electrons come from the donor, in this case, EDTA. Because of that, this is a Lewis acid-base interaction where the ligand is donating electrons like a Lewis base, and that metal ion is accepting electrons like a Lewis acid. You're also going to be thinking about ligand strength in this lab, but it's a little bit different than what you might be thinking about from Chem 106. In Chem 106, you talk about ligand field strength. This is like the spectrochemical series and ranking all of the ligands based on how much they're going to split the d orbitals. And this has to do with the ability of that ligand to donate electrons. The less electronegative it is, the better it is at donating electrons. So that means it will have a stronger field strength. So those are strong field ligands. In this lab, we're not really concerned with strong and weak field ligands, but we will be talking about ligand binding strength. And this is different. It's related to the magnitude of the K of formation for the complex ion. The larger Kf is, the stronger the binding is. And this is related to chelation and also a little bit to ligand field strength. If you have a stronger field ligand, it makes a more stable bond, so we expect the K to be larger. But the big difference here comes when you have a chelating ligand, meaning that it has more than one place where it can make those coordinate covalent bonds. And the reason is because then the molecules will be released. The other molecules, like water molecules that were there before, get released, and that dramatically increases the entropy of the system and makes it very favorable. So for example here, we have some oxalate ions over here on the right that are bidentate ligands versus just water molecules over here on the left. And even though they have about the same ligand field strength, our oxalate complex is considerably stronger. It has a much higher Kf. So the binding strength is much larger. And the reason for that is because now there are only 3 molecules that are constrained instead of having 6 molecules constrained here. so a dramatic increase in entropy when we form that complex with a chelating ligand. In this experiment, you're going to be looking at two different ligands. One of them is called Calmagite, and this shows the Calmagite complexed with magnesium, and it's a tridentate ligand here. So it makes 3 separate bonds with our ions. And when it's in this situation, it will be a red color. So that's like the beginning of your titration when you have all of those free metal ions in the solution making the water hard. At the end of the titration, once the indicator is released, because we get rid of all of the magnesium and calcium ions, it will change to a blue color. And the way we get the indicator to be released is we add something stronger, which is our EDTA. Because EDTA makes 6 ligand metal bonds, or 6 coordinate covalent bonds, it's hexadentate. It's an even stronger ligand. It has a stronger binding strength than Calmagite. And so as you add EDTA slowly, it will slowly gobble up the ions in solution, and eventually all of our Calmagite will be released and it will appear blue. In this experiment, one of the calculations you're going to be asked to do is finding the parts per million of the calcium in your hard water. And it's important to know when you do this that parts per million is the same thing as milligrams per liter. So because a liter would normally be 1,000 grams, then if you go to milligrams, which is there's 1,000 milligrams in a gram, then that's 1,000,000 different from the liter. So milligrams per liter is the unit that you're going for. So you're just going to convert from the amount of EDTA that you have. You'll use the concentration of EDTA to get moles. And you know there's 1 mole of EDTA for each mole of calcium in your sample. You can figure out how many moles of calcium you have in your water sample based on how many milliliters you used. And then you can convert that to grams and then milligrams and turn your milliliters into liters, and you get milligrams per liter for your calcium carbonate. Safety this week, the Calmagite solution can stain your skin, so you should be especially careful of that. And you shouldn't eat or drink anything in lab like the hard water samples that you have. Also this week, the TA evaluation and course evaluation are open, so please do those on Learning Suite. We always appreciate your honest and productive, constructive feedback that you give on your evaluations, and we read those every semester to try to improve the course. Thanks for listening, and good luck in lab.